So my laboratory commonly uses P10 immunohistochemistry to demonstrate the expression of P10 in tissues. So I just want to take you through a typical experiment and show you sort of what the data looks like. So this is a Z-stack from a confocal microscope. And what we've done here, now you can see in the red and the green channel, is we've injected 2 retroviruses into the brain of a P10 floxed animal. And these retroviruses express 2 different things. So this red one expresses mCherry T2A Cre, and so this one should basically cause the deletion of P10 in all of these cells to show us P10 knockout phenotype. Okay, and we co-inject this with a green virus, and so this one expresses just GFP, and so cells that are only green are essentially control cells here. And the first thing you'll notice is that the cells that are only green have relatively modest soma sizes, not very large somas, whereas the cells that are green and red have really large somas. Okay, and this is a typical characteristic of a P10 knockout neuron. Another thing is that the cells that are green and red usually have multiple dendrites coming off of that soma, so they have multiple basal dendrites. Whereas all of these cells that are green only, you're going to see only one dendrite coming off of that cell. So basically, morphologically, already you can essentially identify cells that are P10 knockout versus control cells. And again, this is dentate granule tissue, and this is a retrovirus. So when you inject a retrovirus into the dentate gyrus, it only labels dividing cells. Okay, and the only dividing cells in the postnatal animal are these granule neurons. And so all of these granule neurons are actually the same age. So every neuron labeled here is about 21 days old. And so there's really no variability due to cell types or other things like that. These are all 21-day-old granule neurons. Okay? But in addition to the morphology, it would be nice to have some way to prove that those cells that are expressing Cre are also knockout for P10. And that's where the immunohistochemistry comes in. So I'm showing this here in gray instead of blue just because it's really difficult to see the blue. And so what we've done here is we use the cell signaling antibody. This is a rabbit antibody against P10 from Cell Signaling Technologies. And we've had to use antigen retrieval. Okay, so you have to use heat-mediated antigen retrieval to get good P10 immunohistochemistry, and that does change the quality of the tissue. In fact, the heat-mediated antigen retrieval is why the staining for the mCherry here doesn't look great. It essentially destroys all endogenous fluorescence, and so the only signal we have left here is the antibody against mCherry, which in this case is a mouse monoclonal, which isn't actually the best mCherry antibody. The rabbit monoclonal against mCherry is a rabbit polyclonal is a much better antibody, but we can't stain for P10 with that one. And so the quality of this staining, you have to sacrifice it to get really good staining for the P10. Okay, you maintain relatively good staining for the GFP here because this is a chicken antibody, and it is just a beautiful antibody with very little background. So it still looks very good after antigen treatment. Okay, so I've digressed a little bit. Come back to this anti-P10 staining. So essentially, P10 is in all of the granule neurons, and you can see it out here in the molecular layer because it's expressed in dendrites. This is made up largely of dendrites of the granule neurons and also axons of interneurons in this inner granule cell layer. You can't see the outer molecular layer in this one. You can see also positivity here in the hilus, and you see a couple places in this image stack where there's a lot less P10 staining. Okay, so and I would argue the most important one would be in cells like these. And what you can see is that if I turn on the other colors, that these large blank sections where you don't see P10 are virtually invariably cells that are red, and so these are cells expressing Cre, and so you can see um, quite easily that the M-cherry T2A Cre has caused a reduction in the staining intensity for P10. Okay, that's not to say that there are no Cre negative cells that look like they don't have P10. There are areas in this tissue where it looks like there's a lack of P10 staining, and that is not due to Cre-mediated recombination. Okay, so for instance, this right here is probably just a blood vessel. This is essentially just empty space within the tissue, and those blood vessels look negative for P10. Okay. Down here, though, is an interesting case because I don't believe that all of this is just empty space. There are some different cell types down here. Specifically, there are some precursor cells, and it does appear that maybe some of those cells, which are not red, have lower levels of P10 expression. Okay. However, all of the cells that are labeled with our retrovirus are 21-day-old cells, and it's pretty clear that anywhere there is a loss of P10 expression is somewhere where it's a red cell. Okay. So conversely, if we look at the green channel, so again, cells that are only green. Okay, so we have quite a few instances like this small cell here, green cell, only green. You can see if I turn off the red, there's no expression of M-cherry in that cell. And then if we look at the immunohistochemistry, you see quite clearly that that cell maintains expression. So it's not that virally labeled cells lose P10 expression, and it's not that 21-day-old cells don't express P10. So let's look for another example of that. So it's easier to see if we just turn this off. Now we come through here. So again, this is a cell that's green, and if I click the green on and off, you can see quite clearly that it's not red. Okay, and then if I bring on that, you can see that it again is maintaining P10 expression. And this cell right next to it, okay, so there's cells all throughout here, so right next to it, this is an uninfected cell. It also has P10. And you might notice, see the nucleus here is actually has a little bit less P10 staining than does the cytoplasm. So that's very typical in virtually every cell. You're going to see, again, the cytoplasm is a little bit darker. The nucleus or the cytoplasm is a little bit brighter, i.e. it has more P10 than does the nucleus. But there is, in fact, P10 in the nucleus, because you can see here in a cell where P10's been knocked out, that cell is much dimmer than the nucleus of a neighboring cell. Finally, you see cells like this guy. Okay, so that one you're saying, well, that one doesn't look very red, but it looks like a P10 knockout. And what in fact that is, hopefully you can see this, is it's actually a very dim red cell. Okay, so there's just a very light M-cherry expression, and so I think basically that what this is even in cells that have very low level of expression of the virus and even have very low levels of Cre, that the amount of Cre that's expressed would still be sufficient to cause the floxing of that allele and the knockout of P10. And that particular cell actually happens to also express GFP, and you can see that it is actually, in fact, a very large cell. So, if I can't convince you that it's red, you could say, well, potentially this is a cell that is endogenously missing or lacking P10. And if so, it's quite interesting that it is much larger than any other neighboring cell that looks like it's only green. Okay, so like these cells, for instance. But in reality, and hopefully you can see that in this video. If I click this on and off, you'll see that that cell does in fact have a very little bit of red staining intensity in it, even though it's dim. So hopefully, taken as a whole, you'll now have a better appreciation for what P10 immunohistochemistry looks like from dentate gyrus brain tissue, and you'll be convinced that in cells that are expressing mCherry and T2A Cre, that the immunohistochemistry indicates that we are indeed knocking out P10 in those cells using Cre-Lox technology.